Good day everyone, my name is Nicole Galcipepa and I will be presenting the efficacy of Curcuma Longa and Zingiber Officinale extract as antibacterial agent against Streptococcus mutans. Oral hygiene has a great contribution to the overall health and well-being of a person. Improper oral hygiene may lead to dental infections like dental caries, gingivitis, and periodontitis. Among them, dental caries is considered to be the most prevalent infectious disease in the world. Dental caries is not caused by a single organism but can be composed of several hundred of species. One of the significant contributor of dental caries in humans is Streptococcus mutans. Streptococcus mutans is a cariogenic bacteria found in dental plaque, same for oral diseases and periodontal diseases. But this bacteria may be effectively controlled by maintaining the oral health in patients through mechanical plaque control and proper oral hygiene, which can be achieved through toothbrushing, flossing, and the use of mouth rinses. Either chemically made such as chlorhexidine gluconate and polyhexamethylene biguanide hydrochloride or from naturally occurring plants with antibacterial properties such as Abotilum indicum, Emblica officinalis, Curcuma Longa, and Zingiber Officinale. The research aims to determine the antibacterial efficacy of the essential oils of Curcuma Longa and Zingiber Officinale against Streptococcus mutans. Through observing and examining its antibacterial activity, this has the possibility to provide organic mouthwash which can be used in therapeutic applications. Dental caries is an irreversible disease caused by microbes and is considered to be the most prevalent infectious disease in the world. Systemic conditions like cardiovascular disease, diabetes mellitus, and cancer are more studied and focused in medical society, but tooth decay quietly continues to be prevalent worldwide. Dental caries will not just cause tooth damage, but will eventually lead to tooth loss, which in turn can also lead to malocclusion, malnutrition, social problems, and later to other systemic conditions. Multi-drug resistance and toxicity caused by higher doses of drugs led researchers to explore for alternative substances to fight bacteria. Plant essential oils have been one of the major candidates against pathogenic bacteria, for they are reported to have inhibitory potentials against bacterial strains. Essential oils can be bacteriostatic, meaning it inhibits the bacterial growth, or bactericidal, in which it kills the bacterial cells. They are lipophilic in nature, meaning it can easily enter the cell membranes of bacteria, making the membrane permeable, causing leakage of ions and cellular components, thus destabilizing the bacteria. Curcuma longa or turmeric is one of the most common spices and used as a flavoring and a medicinal agent. Moreover, it has been used as a treatment for different disorders. The chemical responsible for its activity is called curcumin. It is the most active constituent and most important portion of the plant and can be extracted from the plant's essential oil. It acts as a potential source of new drugs with anti-inflammatory, hypocholesteremic, choleratic, antimicrobial, insect repellent, anti-rheumatic, antifibrotic, anti-venomous, antiviral, antidiabetic, anti-hepatotoxic, as well as anti-cancerous properties. Zingiber officinale is a medicinal plant used all over the world for a wide array of unrelated ailments including arthritis, cramps, rheumatism, sprains, sore throats, muscular aches, pains, constipation, vomiting, hypertension, indigestion, dementia, and infectious diseases. It has a direct antimicrobial activity and thus can be used as treatment for bacterial infections. For the research, an experimental design was used to determine the antibacterial activity of Curcuma Longa and Zingiber Officinale against Streptococcus mutans with 12% chlorhexidine as a positive control and sterile water as a negative control. 3 kilograms of each fresh matured plant parts with no signs of decay were collected, then dried using the multi-commodity heat pump dryer designed by the head researchers of the Research Center of the Adventist University of the Philippines prior to its extraction method. For the extraction of essential oils, the raw materials were carried out at the Department of Science and Technology. Streptococcus mutant strain was used for the examination of the antibacterial assay. It was identified by the National Institute of Molecular Biology and Biotechnology following the correct identification characteristic that was provided by the Institute prior to the isolation. For the screening of antibacterial activity of Curcuma Longa and Zingiber Officinale, the method chosen was agar well diffusion method. Essential oil extracts were brought to the biotech for the antibacterial susceptibility testing. 5 replicates were prepared for Streptococcus mutans, and data were expressed as mean standard deviation. Essential oils were used in pure concentration for the testing. Table 1 shows the mean zone of Curcuma Longa and Zingiber Officinale oil extract, positive control, and the negative control. The values presented were inclusive of the 8 mm diameter of specimen cylinder cup for testing against Streptococcus mutans. In order to determine the zone of inhibition alone, the diameter of the specimen cylinder cup should be subtracted from the reported values of the mean zones of the inhibition. Following the United States Pharmacopoeia Biological Reactivity Test, the results were interpreted using the following ratings: 0 as none, 1 as light, 2 as mild, 3 as moderate, and 4 as severe. The result indicated number 2 reactivity rating or minimal zone of inhibition from the 2 extract sample against Streptococcus mutans. This table shows the statistical difference between the group means as determined by one-way ANOVA. Since the p-value of the following is less than the alpha value of 0.05, it implicates that there is significant difference between the inhibition of chlorhexidine as a positive control and those of Curcuma Longa and Zingiber Officinale, but minimal significance between the two plants. As defined, Zingiber Officinale and Curcuma Longa are one of those herbal plants which are commonly used as spices in different countries of the world. Their rhizomes, which are commonly used part of the above-mentioned plants, are filled with secondary metabolites and chemicals that served as source of their effectivity as considerable antibacterial agents. Moreover, among Curcuma Longa and Zingiber Officinale, the study showed that Curcuma Longa extract are more effective than Zingiber Officinale as antibacterial agent against Streptococcus mutans by 0.5 mm. Efficacy may be considered minimal, but it inhibited Streptococcus mutans. The advancement of Curcuma Longa and Zingiber Officinale extract induces a decrease in development of the bacteria. The result of the study proves that the use of Curcuma Longa and Zingiber Officinale as mouthwash can be an alternative from commercially produced chlorhexidine for its benefits in terms of its availability, efficiency, and especially its function in preventing the further growth and inhibition of plaque bacteria. As concluded from the study, Curcuma Longa and Zingiber Officinale essential oil extracts contain minimal antibacterial potential. These two plants are suitable and considerable among ethnobotanical and ethnomedicinal plants due to their antibacterial activity against Streptococcus mutans. The study needs further investigation towards the ability of Curcuma Longa and Zingiber Officinale to inhibit the growth of Streptococcus mutans, and continuance of the study could be done, such as conducting the antibacterial test against oral pathogens by means of prepared mouthwash to test its effectivity.